you. Thank you, Sam, for the introduction, and thank you everyone for coming today. Today, I'm going to talk to you a little bit about work we're doing under a New York State Department of Transportation task order to track the spread of spotted lanternfly using eDNA. So for those of you unfamiliar, and it was great to hear eDNA get mentioned quite a few times during those summaries. Environmental DNA is basically genetic material shed by living organisms into the environment, and that material can be used to detect a target organism. DNA can be found in water, on surfaces, and so we use a couple different methods to collect those samples to detect the organisms. And eDNA can trace its origins back to forensic science, and it was originally used in soil microbial work, then aquatic settings, and our lab is really trying to bring it onto land and use it in terrestrial settings. And so eDNA is, just growing rapidly over the past couple decades. As we see, the number of publications has exploded, and this plot doesn't even include last year's publications. More and more it's being used for biomonitoring in aquatic settings, but around the world, more and more it's starting to be used in a regulatory sense as well. One of the great things about eDNA is you can avoid the pitfalls of traditional sampling that's destructive, which often kills the organisms you're trying to protect, notwithstanding the invasive species. And eDNA provides this data with little to no modification of the environment. We can go out there, collect a water sample, roll a tree, spray some foliage, and there's almost no impact on the environment. eDNA also reduces the risk to survey crews. So the longer people are out there surveying, they're exposed to the elements, they might be put in precarious situations. eDNA puts the survey crew out there for a short amount of time and make sure that they can collect their data quickly. And that signal that you collect from eDNA is often something that's accumulated through time. And so it's not just a snapshot, it's what's been going on in the environment for some time. And so before we really get into the nitty-gritty about eDNA, I want to talk a little bit about the cost of inaction and basically this invasion curve. You may be familiar with it, but basically as time increases, the cumulative cost and cumulative invaded area of any invasive species increases. And so what we want to do is we want to detect an invasive species before it gets to the point where only local mitigation is possible. And unfortunately, most of the time when the word gets out there, it's often at the point where eradication is difficult. And so the earlier we can detect it, the better. And so what eDNA aims to do is basically move that detection earlier in time. And so reduce those costs and maximize the likelihood of, controlling an invasive species. And these detection lags, the time between the initial establishment of an invasive species and its initial detection, it's a really big problem because often when you first have an invasive come in, it's very rare on the landscape. And so traditional techniques to detect an organism don't really work all that well. And so when you're out there surveying, if you fail to detect it because you're using a tool that has a low detection probability, then this is a huge lost opportunity because you think it's not there when it actually is. You may move on to another area. And so you miss it when you could have begun to control it in that area. And so our lab recognized that, the eDNA is really ripe for use with invasive insects. And so what we aim to do is in a number of settings, use eDNA to detect insects when they're often difficult to find with traditional methods because they're small or they have hidden or cryptic larval stages. And so we've begun using eDNA in a biosecurity sense, making sure that an invasive is not moving in through a port or an airport. It can be used to detect satellite populations that are apart from others. Maybe that insect has been moved, through human activities. And it can also be used to delimit an existing population by setting up a transect and then sampling along that transect. And it's also really helpful for relative abundance because we can sort of zoom in on the genetics of a specific organism. If there's, say, a certain biotype that's resistant, you could, in theory, design an assay that would detect just the resistant one, so you can better understand what's going on within the populations. And so our current detection toolbox for invasive insects, well, we have visual surveys, we have roadside surveys. So these are commonly done. They're an integral part of the detection toolbox, but they often detect things like spotted lanternfly when they're already at pretty high density levels. And so, once they're in an area or on a crop, it's not too late to do anything, but you're already behind the curve. Traps are also an important tool that I heard a lot about on the introductions, and they're also critical, but they're passive. They need to be checked regularly. They need to be maintained. And so, there are some problems with them. And then one of the, I wouldn't say newer, but newer to be rolled out technologies is using dogs that can detect invasives. And they're very sensitive to detect them, on par with environmental DNA. But training these dogs can be labor intensive, they're expensive. And as anyone who's worked with these dogs knows, they can only really work for a couple hours a day before their detection probability really drops or they get distracted. And so eDNA can be used sort of alongside these different technologies. And one big question we often get is, how do I know the species is present at a site based on the eDNA sample? What actually goes on under the hood? Well, all of our field aggregation techniques aim to concentrate the eDNA sample onto a small filter. And so that's what you see here in this picture. Contamination, which I'll talk about in a little bit, is always a concern. So you'll see a lot of us wearing gloves in all of these photos. We want to make sure we're not contaminating the samples between sites or from something we may have brought in ourselves. And so once you have the environmental sample, it's all about preserving that sample, bringing it back to the lab, and then extracting the DNA from that sample. Once you have that extracted DNA, you can do a number of things with it. But the two typical pipelines that we use in our lab are QPCR, which is single species detection, or next generation sequencing, which can detect a whole suite of species. So QPCR, it's the same thing that's been used to detect COVID for the past few years. It's extremely sensitive. It can detect as few as a couple copies of a target sequence, a reaction volume. But as I said, with next generation sequencing workflows, you can detect an entire group. And so, it's a little less sensitive, but it gives you a wider net that you can cast within an environment. And all of these technologies rely on basically these genetic markers. We typically use the mitochondrial genome. Can you guys still hear me? I'm getting notices that my connection's unstable. Great. So within the mitochondrial genome, you have all these different regions that you can select from. And so what you're looking for is a unique sequence that you can use to detect an invasive species. So the underpinnings of all of these technologies are really the polymerase chain reaction, or PCR. You've probably heard that in your science classes. And all it really is designing a set of primers, which are just a series of base pairs for your target. And then you put that into the reaction, you run it through many cycles, and at every cycle, you're effectively doubling the amount of DNA of your target. And so you just want to amplify the amount of DNA in your sample to the point where you can detect it with a given instrument. And so QPCR, which I mentioned before, is this PCR reaction with the addition of a fluorescent probe. And so that probe also has a unique nucleotide sequence that matches your target. And so when you amplify that DNA with the probe present, you basically generate fluorescence in your instrument. And so you can actually measure the amount of DNA within a sample and you know whether or not it's there. So beyond presence absence, you also get relative quantity. And so this is a basic output from a QPCR run. And this sort of underpins the work that we're doing for spotted lanternfly. The field work, as I'll get into in a few minutes, is quite simple. One of the trickier parts of utilizing eDNA is really developing an assay that works well and you can trust. And so in this example, we've got 5 different species of bivalves, and we're trying to target the center one, the Chinese pond mussels. And this is work we've been doing in New Jersey in an aquatic setting. And so when we design this assay, we want it to pick up not only, some individuals, but all individuals from the Chinese pond mussel population, but we want it to exclude these other species. And so what you need for that is basically samples or sequences from your target and your non-target species. So you can design an assay that picks up all the target, but none of the other ones. So another question we often get is, what does eDNA sampling look like? Well, it depends on what you're trying to do. But in a terrestrial setting, our roller sampling looks a lot like these 3 panels. And so what we'll do is we'll send out a trained technician with a cleaned paint roller, and they'll roll the surface of a set of trees at a site. And then once that roller's been, dirtied up by what's on the tree surface, we'll take that roller, put it into a sterile bag, add DI water, shake it for 30 seconds or so, and then we remove the roller and the sample's left behind in that DI water in the bag. And so we maintain a very clean environment while also being able to have that sample in that bag. And then what we do is we filter that water containing the sample through a very small pore size filter, and then that acts to concentrate the environmental sample onto that filter. We preserve that filter, bring it back to the lab, and then we can put it into that pipeline that I outlined. And the nice thing about this sampling technology, or the way of sampling like this, is that you can use, mostly equipment that can be bought at a local hardware store. It's a pump sprayer, a roller, small paint rollers, some bags and tubing. The only real specialty equipment needed is a field pump. They're about $1,500 or so, and then the filters. And so, this can be deployed at scale fairly inexpensively. And eDNA sampling is super adaptable. We've used it in a number of settings along the East Coast, as far as Hawaii. And, you can use eDNA to detect invasives in water. You can use it on surfaces like trees, and then also within crop settings. And, the barrier to starting to sample with eDNA is quite low. We can train people in, an hour or so to do this work fairly competently. And it's very accessible. And another nice thing is you can bring it out to the field fairly easily. You only really need the rollers and the sprayer and the pump. And so we used to like to joke that we could bring it around in the back of a Prius or on some golf carts. So, talking about eDNA, another really useful tool is risk mapping. And so, risk maps, nothing new. eDNA, it's nothing new. What our lab is aiming to do, though, is bring these 2 things together. And so they help inform and empower one another. We have a risk map that we start with. We go do eDNA sampling, and then we use that to help refine the risk map and also then inform where we should be doing our eDNA sampling. And spotted lanternfly are a great species to do this because they're sapsuckers, as most of you probably know, and so they produce copious amounts of honeydew, which is rich in eDNA. And so they literally sprinkle their DNA all over the surface of trees. And so by taking a paint roller to that tree, we can easily collect samples and detect them at very low levels, even when you wouldn't see them in a visual survey or even when you're trapping them. And so we really calibrated this work in vineyards and trees in New York or New Jersey and Pennsylvania. And so last year we started to roll it out at scale within New York State. And so when we started planning for our survey, this was sort of the map that we had of New York State as of the end of September. And so we had populations going, up to here. There were a couple areas that, there was infestation present, and then a couple single detections. Our goal was really to design a survey that got out well in front of the invasion front, so like that we could really delimit where that front is. And so we decided, as I mentioned, to use our roller survey technology. And so we have here a roller in a bag from a field site. And so using those detections and that information, we also looked at a risk map that was generated based on highway traffic and the presence of host trees, in this example, the tree of heaven. And so we really wanted to hone in on where we were probably likely to encounter SLF. And so we set up a series of sampling points along highways, at rest stops, tech stops, and truck stops where we think that SLF might be hopping off of vehicles that are moving up and down the highways. And so the sites that we set up were about 10 miles apart, and we had 4 transects totaling more than 400 miles. And so we went out there late summer, early fall, and we rolled tons of trees across 45 sites, and we, washed down those rollers, filtered them down to generate those samples. And, a concern is always contamination. Are you bringing DNA from your target from one site to the other? So what we do to protect against that is we actually generated a negative control sample at every single site. So as soon as we got to the site and set up, we used a clean paint roller, generated a negative, then we conducted our sampling. And then at the very next site, again, we started with the negative control. That way, if we had, DNA on us from site to site, we'd know, and we'd be able to pick that up in those negative controls. And so these were the results from our survey from last fall. We detected spotted lanternfly eDNA at 41 out of the 45 sites that we visited, and we had visual detections at only 3 of the 45, and those are seen here in these magnifying glasses. We had no signs of sample contamination within the field, and all of this work was from 4 days in the field, myself and another researcher within our lab, and spending lots of time driving around the state. We got to know it quite well. We brought those samples back to the lab, and then our lab technician and myself worked to process all those samples, and that work took about a month to generate this data set. And so we see that we have detections, from Newburgh all the way up to Albany, Half Moon area, from Binghamton all the way up to Syracuse, from Binghamton out to Bath, with some lack of detection at a couple sites. And then along Long Island, we had detections all the way out to Orient. And so, as you may remember, the SLF map wasn't this filled in September, but then, this is the map as of December, and it lines up pretty well with the other detections that were eventually made through these other methods. So what is the value of eDNA in managing SLF? Well, it's about getting to risky places for early detection, and then using eDNA to make sure we're not playing catch up and making sure that we're pre-planning for on-the-ground actions. So it's all about, again, minimizing that detection lag. So some take-home messages from year 1. Even though we thought we were going to be able to get out way in front of the invasion front, we weren't even close to it. In some cases, they were 100 miles ahead of where we thought they were. So we're going to try again this year, going all the way to basically the Canadian border in the north, and then all the way to Buffalo in the west. We realize that we can use this to find satellite populations. And the question is, do these coincide with the mapped at risk locations? In many instances that made sense, but it lined up well. But in other instances, we were finding them where it was a low risk on the risk map. So we can use these to help inform and basically improve the risk maps. Another point of evidence, this is another point of evidence that visual surveys have poor detection power. We conducted visual surveys. Didn't mention this, I'm sorry. We we conducted visual surveys after we did all of the eDNA detections. So we looked for 5 minutes at a set of 3 trees twice at each site, and we only found spotted lanternfly at 3 sites. Traps performed a little bit better. There were some SLF in traps, but not much better than the visual surveys. And so we think that this work from last fall provides operational proof that eDNA surveys can be a cost-effective way of surveying a large spatial area over 400 miles in a very short period of time, basically, 4 days in the field and then a month in the lab. And so, what I want to stress is you should, if you're not already using eDNA, you may want to consider adding it to your management toolkit. It can be used in a number of settings beyond what I've presented here. We've used it to sample bulk material for the presence of larvae in Hawaii. So taking the mulch that the coconut rhinoceros beetle larvae grow in, we can rinse that and then filter that down to detect them. It can be used to confirm individual tree infestations. A graduate student in our lab has used it to detect emerald ash borer by coring trees. And it's also been used to detect the persistence of biocontrol insects after they've been released. And so eDNA to some may seem like the future already, but we're still not even on the cutting edge. Some labs are using it to detect species in the air. So they're basically filtering large volumes of air onto a filter and then putting it through the same workflows to describe what they're finding within the air. And some of the latest publications claim to detect eDNA at a continental scale. And then drones are also increasingly being used, especially in the terrestrial realm, to visit higher sites that might be dangerous for people and also just make it quicker to collect those samples. And, many of you probably know McCoy's tricorder from Star Trek. Well, we're basically there. There are mobile battery-powered QPCR instruments like the Franklin instrument here. And there are also next generation sequencing units as well, like the Oxford Nanopore. And so these technologies can be used in the field. There's a little bit of sample prep that still needs to be done, but instead of bringing everything back to the lab and waiting weeks or months, it's possible that you could get same-day detections, not at the same scale as you would if you were sampling a lot and then bringing it back, but at site-specific locations, you could generate some answers with eDNA. And so I want to leave you with the message that there's never going to be a single tool that solves any given invasive species issue. It's always going to be about choosing what works in a given context based on the operational capacity of your organization, a species' life history, the potential impacts of that species, and regulations on that species. But eDNA is a new tool that we can use in our Swiss Army knife of invasive species management. And so by combining risk maps and eDNA surveys, we can more rapidly conduct proactive actions. And there are other uses for this in port and onshore inspections, and also to confirm eradication success. And with that, I'd like to thank all the folks that have helped us with this work over the years and have funded us bringing eDNA surveys onto land. And with that, I'll take any questions.